The overall goal of this procedure is to generate consistent and reproducible airway neutrophilia by exposure to aerosolized LPS. This is accomplished by first loading LPS suspended in saline into a nebulizer connected to an exposure chamber. Next, mice are exposed to the aerosolized LPS, and then bronchoalveolar lavage is collected and inflammatory cells in the lavage are enumerated. Finally, the lung tissue is fixed in formalin for embedding and sectioning and immunohistochemical analysis. Ultimately, the total and differential cell counts of the bronchoalveolar lavage can be used to assess neutrophil recruitment to the lungs.
The main advantages of this technique over existing methods, like intratracheal LPS administration, are the even distribution of aerosolized LPS throughout the lungs, the ease of performance, and the minimal training required for successful application.
To generate an LPS aerosol, using proper PPE and within a ventilated level 2 biohazard hood, begin by inserting a red inlet into a nebulizer and then connecting the nebulizer to pressurized room air via the tubing provided by the manufacturer. Next, use an air filter to connect the outlet of the nebulizer to a mass flow meter and then connect the mass flow meter to an electrical supply. Adjust the airflow to 5 liters per minute, keeping the pressure at 1 to 2 bar, and then remove the mass flow meter and disconnect the airflow.